Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Best Practices for Cell Culture, presented by Mr. Stephen Budd. Mr. Budd is a product line business specialist at ATCC. In this presentation, Mr. Budd will tap into ATCC's vast cell culture experience and share the best practices for culturing cells that ensure optimal results and performance. He will cover all aspects of successful cell culture, including culture initiation, expansion, authentication, cryopreservation, and transfection. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Steve Budd.
Thank you, Brian. Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and service center in Gaithersburg, Maryland. We're the world's largest, most diverse biological materials and information resource for cells and microbes, the gold standard, which leads us into the topic of today's webinar on cell culture fundamentals. In today's presentation, we're going to start with cell handling tips, focusing on cryopreservation, aseptic technique and contamination, cell authentication, and issues with mycoplasma. We'll then discuss reasons for choosing certain cell types from primary cells to cell lines, and finish with some points on transfection and cell viability. So we're first going to start with some cell and media handling techniques. So to start with thawing, when ready to thaw a vial of cells, the most important factor to note is that unlike freezing cells, thawing cells should be formed as quickly as possible to remove the cryoprotectant from the cells. For best results, place the vial in a 37°C bath for approximately 2 minutes, gently agitating the vial to break up the ice. After removal from the bath, make sure to spray the vial with ethanol for decontamination. Transfer the contents to a sterile centrifuge tube with about 9 mL of complete media, which includes a cell culture medium and about 10% of FBS. When adding the growth media, pipette the liquid into the tube slowly in a dropwise fashion to avoid osmotic shock before adding the final full amount. When done, centrifuge the tube for 10 minutes at 125 Gs, discarding the supernatant and resuspend in 1 to 2 mL of growth media. Place the cells in the desired cell culture vessel with additional media. The main take-home here is to thaw cells as quickly as possible when placed in the water bath. Also note that for most primary cells, it is usually advisable not to centrifuge the cells, as this could be detrimental to the cell health. Simply plate the cells after adding the additional growth media. So after thawing cells, they should be plated in an appropriate vessel, which could be cell culture dishes or flasks. This is all depending on your specific needs. For most cell types, the general accepted practice is to allow them to settle for about 24 hours after seeding. At this point, the cells should be checked for confluency, which is the percent coverage of cells over the surface of the vessel. If expanding cells, the ideal time to cell culture is at approximately 80% confluency. The time it takes to reach this level can vary upon cell types. Primary cells can take several days or even up to a couple of weeks. The diagram gives a good visual of the different growth phases of cells in culture. When first plated, cells enter a lag phase where they grow slowly while trying to recover from the stress of subculturing. Cells then enter the exponential phase of growth. Here, cells will grow exponentially as long as growth surface is available. When this surface is completely occupied, cells then enter the stationary phase of no growth. If they persist in this phase, they will then enter the decline phase, not shown in this graph, but to the right of the stationary phase. At this phase, they will lose viability and essentially die. To ensure phenotypic stability, cell lines need to be maintained in subcultures in the exponential phase. This will need to occur before a cell monolayer becomes 100% confluent or reaches its maximum recommended cell density. As stated before, that ideal confluency is about 80%. So when you've reached 80% confluency, cells can be passaged with trypsin EDTA. Trypsin disrupts the proteins involved in attaching cells to the culture vessel. The figure on the right shows a progression of cells being trypsinized. At the starting point, we see cells attached as a monolayer. After trypsin is added to the cells, they become flattened and more round. When completely detached, they can be seen floating around in clusters. This should take about 3 to 5 minutes, but make sure to note that the dish should be gently agitated to help detach the cells, otherwise it could take longer. Long-term exposure of trypsin to cells can be detrimental, so that time in trypsin needs to be minimized. Also note that if using primary cells, a special lower concentration of formula of about 0.05% trypsin should be used. The lower concentration of trypsin will definitely require the physical agitation of the cell culture dish to ensure detachment. Also, when trypsinization is complete, the reaction needs to be quenched, ideally with a soybean neutralizing solution. Again, this will help protect the cells from any damage of long-term exposure to trypsin. In the next several slides, we want to discuss techniques on cryopreservation. I want to first go over the basic theory behind cryopreservation and how it works. So the freezing process is a complex phenomenon that's been studied for decades. Cellular metabolism stops when all water is turned to ice. This, however, is largely detrimental to the cell's health. Two main reasons for this: ice formation and an increase in solute concentration within the cell. This is caused by either freezing too quickly or too slowly. The solution to this problem is maintaining the ideal freezing rate while using a cryoprotectant, the most commonly used being DMSO. The combined effect will minimize ice crystallization and reduce the solute concentration effects. An important take-home here, as the diagram notes, for most cells, the optimal rate of freezing is a drop of 1 to 2 degrees Celsius per minute. So freezing cells is typically a two-stage process in which cells are incubated for some time at minus 70 degrees Celsius before being stored at minus 140 degrees C. As previously discussed, cells must be cooled at a rate of about minus 1 degree Celsius per minute. This can be achieved with the use of a controlled rate freezer or controlled rate chambers. The length of time needed in the minus 70 will depend on the type of chamber used. So after spending the appropriate time at minus 70 degrees C, the vials must be transferred to minus 140 C for long-term storage. It's recommended to store cells in liquid nitrogen tanks for this purpose. So as I just stated, long-term storage of mammalian cells require that cells be exposed to temperatures below minus 140 degrees Celsius. At these low temperatures, metabolic activity ceases, allowing for the preservation of cells for an indefinite period of time. For storing biological material at these low temperatures, it is highly advisable to use liquid nitrogen freezers. So there are two basic types of liquid nitrogen storage practices. One is immersing the vials directly in the liquid. The other involves holding the vials in the vapor phase above the liquid. Vapor phase systems create a vertical temperature gradient within the container. At the very bottom, where the liquid nitrogen resides, the temperature will maintain about minus 196 degrees Celsius. The temperature in the vapor nitrogen decreases as it reaches the top portion of the container. Enough liquid nitrogen should be used to guarantee that the warmest part of the container at the top is always at a temperature at or under minus 140 degrees Celsius. It is highly recommended that vials be stored in the vapor phase rather than the liquid. Since metabolic activity is arrested at minus 140 degrees C, the additional cold temperatures at the liquid phase offers no real advantage. However, immersing the material in the liquid phase runs the risk that some of the liquid nitrogen could penetrate the vials and cause them to crack or even burst. As we leave the discussion on cryopreservation, I want to discuss cell characterization as it relates to our upcoming topic of general cell health, contamination, and authentication. It is necessary to characterize cells to determine how healthy and viable they are, as well as making sure that they are the cells you think they are. Simple cell viability counts using trypan blue with the hemocytometer are an easy way to determine the percentage of live cells over dead cells. With this method, live cells appear to have bright centers while dead cells appear dark blue as the dye penetrates the compromised membrane. Often when researchers receive cells from collaborators or other unreliable sources, they may find that the cells are not exactly what they thought they were. So morphology is a quick way to distinguish broad cell types from one another. In the example on the right, you have two cell types, fibroblasts and HUVECs. They have very distinct morphology. Fibroblasts tend to be elongated, whereas HUVECs tend to have a rounded cobblestone appearance. Also, since different cell types grow at different rates, always make sure your cells are growing the way you would expect. If cells are contaminated with another fast-growing cell, the growth rate and doubling time may be greatly increased. We now come to the topic of contamination. So the main types of contamination we'll be concerned about are microbial, which include bacterial, mycoplasma, fungi, and virus. There's also cellular contamination, meaning one cell type or cell line has invaded another. We'll discuss this in a little more detail shortly. So bacterial and fungal contamination are the most common problems seen in labs. They often appear quickly, but fortunately can be detected very easily. Turbid and acidic media usually indicate the presence of bacteria. Cell stress from microbial contamination may also lead to morphological changes in the cell type. Fungal contamination is seen by the appearance of obvious filamentous structures in the media. So at least one good thing about this microbial contamination is that it's very easy to detect quickly. Mycoplasma and cellular cross-contamination, discussed in just a minute, is not as easy to detect. So where does contamination come from? For starters, anything that has been contaminated in the past can contaminate cells going forward. Media and reagents that are not handled correctly can easily be a vehicle to introduce bacterial and fungus into cell cultures. Using items such as petri dishes or pipette tips that are not properly sterilized can certainly introduce contaminants. Personnel can also introduce particulates and aerosols on personal clothing or even on dry skin. Improperly maintained equipment can prevent protection from the entry of contaminants. Overpacking autoclaves or ovens can even cause problems with uneven heating, creating pockets of non-sterile supplies. The flow rate of hoods, if not properly adjusted, can allow particulates to enter. So what can you do to make sure cells and cultures do not get contaminated? So I'm going to point out a few tips that should prevent contamination problems. First, in general, you want to make it hard for contaminants to enter your cell culture vessels. Small dishes, such as 35 to 60 millimeter dishes, can be placed in larger ones when in the incubator. Placing plates and dishes in larger ones are also ideal for transport. This adds an additional physical barrier that prevents particulates from entering. When using flasks, it is recommended to use vented caps that can be sealed tightly but still allow gas exchange. I always use disposable aspirators and pipette tips that are pre-sterilized. Make sure that your hood has adequate laminar flow and never overcrowd or leave materials such as boxes or bottles in the hood indefinitely. This can impede proper flow and block attempts at cleaning and wiping down the hood surface. When bringing media and reagents into the hood, always spray the items down with alcohol to remove any contaminants. It is also advisable to bring in small amounts of reagents into the hood at any given time. If possible, you may want to aliquot small amounts in these containers to reduce the number of times you're bringing a possible contaminant into the hood. For instance, if a vial is contaminated, at the very least, it won't continue to enter the workspace. Always remember to wear clean lab coats that cover most of your arms to prevent dust and shedding skin to contaminate your cultures. We advise not using antibiotics in culture. There are a few reasons in addition to the potential of toxicity. If you use media with no antibiotics and a contaminant enters your cell culture, chances are it will be visible within a couple of days. You can then discard the infected dishes. However, if there are antibiotics present, it may slow the growth of the contaminant, allowing further passages and freezing down your cell lines. In short, you may be saving large amounts of contaminated cells that can cause problems down the road. Resistant mycoplasma may also get introduced along with other bacteria. The antibiotics may get rid of the larger bacteria, but mycoplasma may persist undetected. So in short, antibiotics can also sort of cause this sort of sense of false security. When is it acceptable to use antibiotics? So a few exceptions make it necessary. For instance, if you're isolating primary cells from original tissue, it may be a good idea to use the antibiotics for the first couple of weeks. So mycoplasma contamination is a separate contamination issue that is extremely difficult to detect. So contamination from mycoplasma can cause a host of problems that includes altering the cell's function, growth, morphology, causing chromosomal aberrations, affect nucleic acid synthesis, and changes in membrane antigens and gene expression. The main source of mycoplasma contamination is receiving infected cultures from other research laboratories. Reagents purchased commercially can also be a source of contamination. Mycoplasma contamination can be detected through a number of available kits in the market. The Universal Mycoplasma Detection Kit from ATCC is a PCR-based kit that can detect 60 of the most commonly identified mycoplasma species. So to put it in perspective, over 90% of contamination is caused in just a few species. The Mycoplasma Detection Kit comes with specific primers that are unique to mycoplasma with mycoplasma DNA as a control. To test for mycoplasma, simply collect and lyse cells from your culture, perform a touchdown PCR, and run the product on a gel. If the cell culture is contaminated, a band will appear at the same location as a positive control. Cross-contamination of one cell type with another is also a problem. If the contamination is a particular fast-growing cell, the original cell line can be gradually replaced with the invading cells. As a result, an entire cell line in one's depository can be completely overwritten with a new cell. Furthermore, morphology is often not reliable enough to distinguish between the original and invading hosts, particularly if they're the same cell type, like one endothelial cell invading another endothelial cell. So there are a few common causes for this kind of contamination. Researchers can contaminate their own cell lines by working with multiple cell lines at a time. This often occurs when using the same pipette tips or aspirators with vessels containing different cell types. We recommend only working with one cell line at a time under the hood if at all possible. More commonly, researchers receive cell lines from other labs or companies that were already cross-contaminated, so the importance of this cannot be understated. It has been estimated that 20% of scientific publications use misidentified cell cultures. This has also resulted in about 50% of preclinical research being non-reproducible. As a service, ATCC offers cell line authentication utilizing short tandem repeat profiling. STR profiling helps to detect misidentified, cross-contaminated, or genetically drifted cells, which invalidate research results. To take advantage of this service, simply receive a sample packet through the mail, spot cells onto a paper insert, and mail back. You will receive a report that includes the STR profile of your cell line, a comparison of your cells against an STR profile database, electropherogram supporting the allele cause at each loci, and a complete interpretation of the results. I also like to note that when you get back into the lab, now is a good time to start with freshly authenticated cells, and we do now offer human and mouse authenticated cells. So here I'm going to change focus and go into how you can choose the best model. When we're talking about which best cell model and why you would want to choose this. But before you go into that, I want to briefly discuss media before you go into the models. So in general, continuous cell lines use a classical cell culture medium, such as DMM, RPMI, 1640, etc. usually with 10% FBS. More specialized cell types like primary cells, H-church cell lines, stem cells, tend to require more specialized media. It is generally recommended to maintain cells in the same medium, but if transferring to a new medium, it should be done gradually with the 50-50 mix of old and new, eventually reaching the new medium entirely. ATCC does not recommend using heat inactivated FBS under normal circumstances because it can denature growth factors necessary for the health of the cell. There are several types of cell models to choose from when designing your experiment, each with its own set of pros and cons. So let's start with the far right, the continuous cell lines. Continuous cell lines are often derived from a tumor. They're generally very easy to culture and have a low cost of culturing. They do not require a complex growth medium, usually just one of the classical cell culture formulations like DMIM or RPMI supplemented with FBS. Like a tumor, they have great proliferative capacity. They all originate from a single clone, so there's minimal inter-experimental reproducibility. However, they are by nature abnormal cells with high instability and thus provide low in vivo predictability and limited biological relevance. The next model, cell type that I would like to discuss is H-church immortalized cells. These cells are genetically modified primary cells such that the cell no longer undergoes senescence. So what it's created is a cell with continuous cell-like growth characteristics but primary cell physiology. This means high proliferative capacity, and since every H-church cell type originates from a single transfected clone, there's no inter-experimental variability. Every lot will be virtually identical. Since the starting material is a normal primary cell, there is greater genetic stability with near normal primary physiology, so therefore high predictability and high biological relevance. The third cell type is an induced pluripotent stem cell derived primary cell, or iPSC derived primary cell. This cell type is derived from a somatic cell line that is dedifferentiated to an embryonic state and then differentiated to a specific primary cell lineage using a specific cocktail of growth factors. iPSC-derived primary cells are usually generated from a single clone, so there's likely little worries regarding the introduction of biological variability. The main problem with stem cell-derived cells is that they are differentiated in the immature primary cell types, so not the best for predictability or biological relevance. Lastly, we'll discuss the cell type that is the best model for in vivo predictability and biological relevance, the primary cell. A primary cell is usually isolated from normal adult tissue, so no genetic instability. However, for large-scale screening, there is a potential problem, not enough cells from a given donor. The use of new donors will introduce biological variability in your experiments. So each model has its own strengths and weaknesses. However, as you move from basic research to more clinical-like applications, you need to choose a model that most positively matches up to your research needs. As a final note regarding choice of cell model, continuous cell lines are derived again from a tumor or mutated in some fashion, such that the cell has deviated from its original source. So in every experiment involving continuous cell lines, the corresponding primary cell should be run as a control. That way, if you observe a specific effect during your experiment, you can be sure the results are valid and not an artifact of the tumor or mutation. So the take home is use primary cells as controls when at all possible. Okay, we're going to switch gears again and talk a little bit about transfections and what transfection reagents you can use. So now I'm going to discuss the basic method of lipid-based transfection. So transfection is a method for introducing exogenous nucleic acid sequences into cells. There are various ways to accomplish this. One of the most common is lipid-mediated transfection. Lipid-mediated transfection is the most accessible and usually the simplest method available. It's based upon the idea that opposites attract. The transfection reagent is a cationic lipid that forms micelles with a net positive charge. Nucleic acids have a net negative charge, allowing the formation of complexes of lipid and DNA. The nucleic acid is covered by the micelle, so the complex now has a net positive charge on the outside. The surface of a cell has a net negative charge, so the complex moves to the surface and binds to the cell membrane. The nucleic acid lipid complex can now move through the cell's lipid membrane, allowing the nucleic acid to enter the cell cytoplasm. The nucleic acid then enters the nucleus, and the gene product can be expressed. ATC supplies two lipid reagents for transfection, GeneX Plus and Transfex. Transfex is our all-purpose reagent working well with continuous cell lines in primary cells. As suspension cells are a bit more challenging for transfection, we often find that GeneX Plus offers higher transfection efficiency for these suspension cells. So it's very easy to actually set up a lipid transfection process. And here I'm going to go through a simple workflow to demonstrate this. You always want to transfect cells when they're in the exponential growth phase. So you seed your cells to be ready the day before you plan to do your transfection experiments. On transfection day, which is day 0 on the slide, you combine your DNA with your lipid reagent and allow the complexes to form. Then you can add the complexes to your cells that you seeded on day -1. It's at the very top of the chart. You should then be able to detect protein expression at about 24 to 72 hours post-transfection. So now we're going to sort of talk about transfection best practices and how to optimize culture conditions. And these are 4 things to take into consideration. So even though it is a fairly straightforward protocol, there are several aspects of the process that you need to be optimized to ensure the highest potential, the possible transfection efficiency. To achieve a good transfection, the health of your cells is of utmost importance. The cells should preferably be at a low passage number. If they're seeded too high or too low of a density, the metabolic rate could be negatively affected, and cells could find the complex as toxic or may not readily express the gene product of interest. Contamination can lower the observed transfection efficiency, especially if it's mycoplasma. If you ever observe a drop in efficiency with the transfection protocol that has been previously optimized, mycoplasma contamination may be a likely reason. Media used during the transfection process is also a consideration. Certain lipids require no or low serum during the transfection process, so always follow the lipid manufacturer's guidelines. So a little bit more on transfection best practices. So for nucleic acids, which are DNA or RNA, one should use high purity and endotoxin-free material to achieve the highest transfection efficiency possible. Endotoxin appears to lower transfection efficiency. For DNA, one should verify promoter strength for the given cell being transfected. Certain promoters are known to work better with certain cell types. Plasma size is also a concern. The larger the plasmid, the lower the expected transfection efficiency. You should also verify that you are using a complete sequence in your plasmid. DNA plasmids that have been stored for some time can become degraded or nicked. So when designing your transfection experiment, you need to determine what assay methods you will employ at the timing for the data collection. As your protein of interest is expressed, you can use real-time PCR to detect RNA, probably 18 to 48 hours post-transfection. If this is your first time working with proteins, I would suggest running a time course analysis at a few different time points. If you plan to detect at the protein level, you need to give the cells enough time to make your protein and then perform any analysis before the protein is turned over. If you are not sure of your protein's half-life in the cell, again, a time course analysis is warranted. So of course, depending on the nature of your protein, there may be additional functional assays you can run. However, I would always recommend running analysis at the genomic and protein level, at least until optimal transfection parameters have been established. So regarding transfection reagents, there are three main areas of concern. First, which lipid should I use? That would depend on the cell type you're using and the cell characteristics. As I just mentioned, certain lipids are generally geared towards cell suspension, like ATCC's Gen X Plus. Other considerations involve what are you trying to deliver to the cells. Certain lipids are for DNA, others may be designed to work with RNA or small proteins. Next, you often have to optimize the ratio of lipid to DNA for the cell type and substrate that you're working with. Most manufacturers will provide guidelines, but some optimization should be expected. Lastly, one should optimize the incubation time for the lipid in contact with cells. If you're using a low or no serum transfection medium, you do not want to leave the cells in the medium for too long, and some complexes can become toxic to cells. So again, most suppliers will provide guidelines, so optimization may be required for your specific conditions. And so this is the very last slide on transfection, and it's going to summarize best practices on the transfection section of this talk. So to achieve optimal transfection efficiency, use healthy cells in the exponential growth, free of contamination, DNA that is of high purity and endotoxin free, and the recommended lipid for each cell type and substrate should be determined. Every setup may require some optimization, including but not limited to seating density of cells and the ratio of lipid to substrate. And so now we're going to move to the last major topic of this discussion, and this is going to be viability assays. So to cover growth and viability, we want to discuss ways of actually measuring the number of cells in a cell culture vessel and determine the proliferation or growth rate. This is ideal for assays in which you want to compare normal growing cells against an experimental factor that would positively or negatively affect the growth. So these two assays that we offer, MTT and the XCT cell proliferation kits, utilize tetraxyllium salts to deliver a quantitative analysis of cell numbers. So this slide is a diagram that shows how this works inside of a cell. So you can imagine the blue spheres are cells, and the gray inner sphere is going to be the nucleus. So with the MTT assay, the tetraxyllium salt is introduced into the cell. Naturally occurring oxidation reactions within the cell, which is indicative of normally growing cells, will reduce the tetraxyllium into formazine. So the cells are then lysed with detergents, releasing the formazine, giving off a purple color. The more purple the media, the more metabolically active and growing the cells will be. So this assay is good for measuring things like cytotoxicity and apoptosis. And the XCT reaction on the right is very similar, but the reaction takes place at the surface, and it gives off an orange color as an indicator. From a more practical point point of comparison, so both assays are designed for 96-well plates, and here we have an overview of what these assays are going to look like. So for both, they're plated with, in this example, with a cytotoxic test compound. With the MTT reagent at the top, there's an incubation period to allow the reagent to enter the cell. The detergent is added, allowing for the compound to be released in the media. At this point, it can take a few more hours for the reaction to complete. With the XCT reaction, the XCT reagent and activation agent are added at the same time, so the reaction starts immediately. So one just practical benefit of the XCT kit over the MTT is there's one less step in the protocol, which will knock off a couple of hours from the whole process. And so this graph shows an example of absorbance versus cell number when comparing the MTT kit and the XCT kit. The XCT is in the blue at the top, the MTT in the red at the bottom. So in this case, eventually the absorbance will saturate and no longer indicate the number of cells as the number of cells increase in your plate. So the reading is taken on the growth curve. So this shows that with the XCT kit, it's more sensitive than the MTT, and that they can read cells between 5,000 to 30,000 cells. So with the MTT, the reading is between 5,000 and 15,000 cells. So the XCT assay, because of its fewer steps in the protocol and its increased sensitivity, is generally considered to be a more sophisticated assay. And so now we're reaching the end, and this is just one last summary on everything that we've talked about. So as a summary of what we've discussed, remember to use best practices to eliminate contamination while ensuring optimal growth and storage. In your cell culture workflow, choose the best model for your needs. Cell lines for standardization and primary cells as optimal controls that maintain biological relevance. H-turn immortalized cells can be the best of both worlds with continuous growth and primary cell characteristics. And so the key to good transfection practices rely on the use of healthy cells, purified substrates, and optimization of the lipid to substrate ratio. Lastly, you can monitor cell health and viability with the MTT and HTT viability assays. And that concludes this webinar on basic cell culture handling. And I will turn it over to Dr. Shapiro for further announcements.
All right, well, thank you, Steve. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org.
And it looks like Michael Utaro has joined us. Mike is
Hi, how are you all doing?
Mike's one of our technical service representatives. He provides technical support for customers having issues growing a particular cell line, and Mike's got years and years of experience growing especially difficult cells. So welcome, Mike.
Well, how are you all doing today?
Let's go ahead and jump to our first question. Can you identify the mycoplasma species with the mycoplasma detection kit?
Ooh, so it's designed to be sensitive enough to read femto levels of the DNA to indicate the presence of any mycoplasma. However, it can't really identify the exact species because if it was that sensitive, it would miss many other species. So it's generally designed to let you know that there is a mycoplasma contamination, but unfortunately, it is unable to really kind of differentiate between specific species.
Okay. Steve, do you have anything to add to that?
No, other than everything you said is pretty thorough. It is a PCR based, yeah, designed to be either an all or nothing. And so again, the idea is it doesn't really matter what the contamination is, what the micro-contamination is. It doesn't matter what the species is. Recommendation is to discard it regardless. So yeah, it's just an all or nothing. Very sensitive. It's very successful. Very good kit. Yeah.
All right. Good. All right. So this next question kind of goes back to what we were talking about growing cells with antibiotics. Can you use a contaminated culture after treating with the antibiotics?
Generally speaking, it's usually not recommended to use cells in cell flasks after they've been contaminated, just to kind of remove that sort of problem aspect from your incubator, because in addition to it being fairly difficult to completely clear cultures of contaminants, the treatment may have compromised cell function. It could affect cell characteristics. So unless the cells are absolutely vital, we generally recommend that any contaminated culture should just be discarded of and cleaned up immediately.
Okay. So when in doubt, throw it out.
Exactly.
Okay, good. All right, I like this question. What are the main reasons cells may unexpectedly detach?
Ooh, that is a good one. So there's definitely quite a few reasons for cells showing poor attachment in culture. One of the more common ones is going to be suboptimal pH of the medium that's being used. If pH is too high or too low, that can have an immediate effect on how cells want to attach to the flasks. Additionally, buffering systems within the media, specifically sodium bicarbonate, are going to dictate how much CO2 you would want to have in your incubator. And so with ATCC, we like to formulate our media with 1.5 grams per liter of sodium bicarbonate, and that works out best with a 5% CO2 incubator. Other vendors' formulations tend to use 3.7 grams per liter of sodium bicarbonate, and that level of sodium bicarbonate would actually do better in a 10% CO2 incubator. And so a lot of times when people try to use that higher level sodium bicarbonate in a 5% CO2 incubator, that is going to affect the pH of the culture, and that can cause some issues with attachment later on.
Okay.
Yeah, and sort of to add on. Oh, just to add on to that, I was going to say. Also dealing with primary cells, so I like to bring that up. If you primary cells have a finite doubling, usually 10 to 15, depending upon the cell type, 10 to 15 doublings, and keep a note that if you go too far with that, they tend to just simply stop growing, and it will just tend to detach there. So when you're working with primary cells or any other kind of cell type that has a finite doubling, just make sure you follow the guidelines and don't take it further than is recommended.
Is there any effect of exposure or concentration of dissociation enzyme?
That could definitely be something that plays a role. So like whenever cells are subcultured, you always want to make sure that if you're using trypsin, that is being inactivated by serum. Typically, most inactivation mediums will just be something 5% FBS rather, and that will inactivate the trypsin that is used to remove the cells from the flask and kind of prevent them from further cutting that could affect further cell attachment or cell growth.
Okay, good. All right. Here's a question about the cool cell, the freezing container that we provide. Can you keep cells in the freezing container for long periods, like several days or a week? And I assume that means keeping it at minus 80 before putting it into liquid nitrogen.
Ooh, so this is definitely something that it should not be kept in minus 80 for several days. Absolutely not. Cryopreservation of cells can be very finicky, and you definitely want to make sure that you're moving the cells from temperature to temperature at the right time. So the cool cell is designed to freeze cells slowly at minus 7 to minus 80 for 4 hours to overnight, to kind of, slowly bring them down to that temperature to prevent ice crystal formation inside of the cells. Once they reach that temperature in the cool cell, you would want to move them to liquid nitrogen as soon as possible. Otherwise, keeping them at minus 70, minus 80 is going to affect viability of the cells. It's going to rapidly affect viability of the cells if they stay at that temperature for too long.
Okay, good answer. That kind of leads into this next question that we got here. So in all cases, cell culture racks within a nitrogen storage unit, say a dewars, must be above the liquid nitrogen level.
So it definitely shows. So when you store cells in liquid nitrogen, they are typically advised to be kept in the liquid nitrogen vapor phase, which is going to be that vapor phase directly above the liquid nitrogen. It's not necessarily going to be detrimental to cells if they are submerged in liquid nitrogen. The biggest concern there would be any sort of cracks or openings in the vial where liquid nitrogen would be able to seep into it. At which point, when you take it out and that nitrogen starts to turn into a gas again, it could cause explosions of the vials, simply put. So it's more of a safety thing, but it should be cold enough both in the vapor phase and submerged for cells to be stored for a long time, but definitely try to keep it at the vapor phase.
Yeah, I'll never forget the first time I had a vial explode on me after pulling it out of the liquid phase of liquid nitrogen.
Mm-hmm. Yeah, no, it's a lesson you learn once, and you learn it forever.
Right. Speaking of lessons learned, have either of you guys had any experience using mycoplasma removal agents?
I have not actually. This is, again, generally like with what we talked about previously with contaminated cultures, it's generally best practice to just remove that culture to prevent it contaminating any other cultures. There are agents, mycoplasma agents that are available commercially to try and cure some cell cultures that are infected, but of course you always run the risk of affecting cell growth, cell characteristics, because some antibiotics and antifungals can be very harsh on cells, which can affect viability.
Right.
Yeah, and also remember if you do try to go that path, you have to make sure and retest the cells for mica to make sure you got rid of it. But right, yeah, and it's the same. Again, it's only recommended if you're dealing with very valuable cells that are very hard to replace. The recommendation again is always just get rid of them.
Exactly.
Okay, I like it. All right, so we all have experience with UV light being used to kind of keep the microbial load down inside your biosafety cabinet. Mm-hmm. Can ultraviolet light sterilize the medium flasks? I don't know if they mean just the flask or the medium in the flask. I guess we can try to answer both.
For medium, for actual liquid inside flasks, absolutely not. UV will definitely not be able to sterilize liquid media like that unless it seems impractical in that sense.
Right.
For surfaces.
And I was going to say, doesn't ultraviolet light also convert a medium component into something that'll kill the cells as well?
It does. I know that
glutamate or something.
That sounds right, because I know it definitely. I mean, the UV light is going to cause reactions. I don't know the exact mechanism for that, but I know that just the energy from UV light is going to cause reactions. It's going to affect the formulation and makeup of that media.
Yep. Yeah, I think that we can just sort of say, don't do it.
Yeah, I mean, there are like 0.22 micron filters are generally more than enough to be able to remove any sort of contaminants from your media if you want to do that. Filtering would definitely be probably the recommended way to go with that.
Yep. I would go along with all that. Okay, here we go. This is a good question. Can you repeat why it is not recommended to inactivate FBS with heat?
Okay, yeah, this is a good question. This is one that we see very commonly because heat inactivated FBS is still very commonly used. So the reason that FBS used to be heat inactivated was to treat for any mycoplasma contamination that was present in the FBS. Since then, FBS has been being triple filtered through multiple small micron filters to remove any traces of mycoplasma. So the original need for heat inactivation is not really an issue nowadays with most modern lots of FBS. And also heat inactivation just it can have a serious effect on growth factors that you see within the serum. So with heat inactivation, you're heating up serum to, I believe, 56 degrees for half an hour. That's long enough for a lot of growth factors to denature. You may not see the same level of growth promotion with heat inactivated serum as you would with non-heat inactivated serum. So in general, unless it's absolutely needed for your experiment, for example, one common cause for wanting to use heat inactivated serum nowadays is because the complement in the serum can affect cells in the culture. So that would be one of the main reasons people would want to use heat inactivated serum. But in general, the FBS is going to be sterile, free of mycoplasma, and you should use a non-heat inactivated serum whenever possible.
Okay. And this feeds into the next question, actually. So is filtering enough to assure that the FBS is adequate for culture?
Yes, it definitely should. So the FBS that you receive should have already been triple filtered. Pretty much any reputable cell biology site, ATCC sells triple filtered. I'm pretty sure Thermo would as well. But pretty much all FBS is going to be triple filtered to remove any presence of mycoplasma contamination.
All right.
And so
good,
heat inactivation would not be necessary.
Gotcha. All right. Here's a question that I like about a dissociation agent. Do we use any other cell dissociation agents besides trypsin, for example, Accutase?
I believe we do have a cell line that is recommended to be dissociated with Accutase. I would have to check. And see our catalog to see that. In general, trypsin is kind of the gold standard for cell dissociation. Depending on the cells, because trypsin it's a protease. It's going to cleave those proteins that are keeping the cells attached to the flask, but then also depending on the cell, there may be additional targets that this protein would want to cleave. It may be too harsh of an action in general on the cells, in which case other alternatives such as Accutase, other non-enzyme dissociation agents may be worthwhile looking into. But for the most part, 0.25% trypsin should work out just fine with most cell lines. For more sensitive cell lines, there's a lower percent trypsin, 0.05%, that may be a more gentle option to use when trying to dissociate certain cell lines.
Right.
Yeah, and on that note, I was just going to say, we do offer a non-enzymatic cell dissociation solution. And, benefit of that is, if nothing else, these components are animal-free. So if your application needs to use animal-free components, that's a good option. That's another reason why you may want to use that.
Mm-hmm.
Right. Is that the same as our stem cell dissociation reagent, which is it's safe for using with iPSCs?
I believe so. I would have to actually double check that. I'm not sure off the top of my head right now.
Oh, so you were asking that? Yeah, those are two different things. Yeah, similar, but the stem cell was used specific. It's been the stem cell dissociation reagent is specifically optimized for stem cells.
Right, right. That comes from a neutral protease from Bacillus polymixta. That's right.
Right, yeah.
Okay, and one point with when you're going to use this is serum doesn't inhibit its activity. So we actually have something specifically to inhibit it. Okay, well, let's move on to the next question, shall we? I've seen some protocols calling for the addition of ascorbic acid to cell culture media. Do you know of any specific advantages or disadvantages of this additive? And I actually kind of remember doing this. I used ascorbic acid as a part of a medium formulation for growing cells that were primary cells. So they were bovine primary adrenal glomerulosa cells, and the ascorbic acid was actually added to that was a free radical scavenger, actually. So basically it's like why you would add it to anything. I guess preservative isn't exactly the right word for it, but the ascorbic acid definitely kept the cells alive longer.
Yeah, I mean, basically it's a form of vitamin C in a way. So yeah, and it does offer some protection against things, protects like fatty acids and things of that nature. So there's a number of reasons, but yeah, it's protection against radicals. Free radicals is a large function of that, I think.
That's right. Protects esterified and non-esterified unsaturated fatty acids from peroxidation. That's right. All right. Now that was pretty important because that was a lipid synthesis project that I was working on as well. So,
nice.
Okay. way back in the day. All right. Here we go. How do you control for the toxicity of the electron transfer reagent required for XPT and other water-soluble petroleum reagents?
That is a good question. I would actually have to look into that a little bit. I wouldn't know that off the top of my head, unfortunately.
Okay, no, that's fair enough. We can definitely look into that and get back with you. I mean, one of the things with that is that you're going to be adding the XPT reagent to all of the wells in that particular experiment, right? So even the control wells are getting the same thing. Presumably, it would have the same effect across the board. But that's just me taking a stab at it. All right. How often do you recommend testing cell lines for mycoplasma, parentheses, that are in heavy use?
That's a good question, actually.
Yeah, one thing that I recommend it would definitely be when the cells aren't performing correctly, right? When your positive control isn't working and when your negative control isn't working, just for starters. But how often else would you say?
I mean, if they're growing... Oh, go ahead. Yeah.
No, let's go. You go ahead. Go ahead.
Oh, I was just going to say, if they're growing, continuously in culture, at least, every few months, I would say, at least, once a year. But if they're growing continuously, you have them just expanding, every few months, I would probably suggest testing for it. Or, and of course, if there's any reason that you suspect you may have had contamination or things, like Brian said, if things start looking funny or whatever the case may be.
No, that sounds just about right. Like especially if you're going to be doing cell culture continuously, every few months or basically whenever a need arises where you feel like for your own state of mind, or if projects aren't growing as expected, testing as needed and every couple of months.
Yep, that all sounds good to me. Okay, here's an interesting question about record keeping. What is the best labeling method for keeping track of passage number and dish number from cells split from the same passage?
So it's definitely going to vary pretty drastically lab to lab to lab. I could definitely say that. The most important thing is that anybody in your lab who looks at a flask will be able to kind of like immediately be able to see it's this cell line at this passage, it was started at this date, and it's being grown by this person. Paper trails can help with that. I mean, having a logbook of, when you start a vial, I've seeded it into this flask, all of this was handled by me, keeping passages written on the flask in your written notes, basically just kind of keeping as much information as you can so that anybody who is working in the lab will be able to look at a flask and be able to understand, who is responsible for that flask.
Okay. Yeah, I definitely recommend some form of record keeping. You might want to use a decimal point to separate passage number from which original stock you're working from. Might be a good way of going about doing that. I did that in a lab once. Okay. So first of all, this asker says, "Nice speech, guys." But they just wanted to confirm, don't heat inactivate FBS, but then about thawing. So should you be thawing the FBS directly in a 37-degree bath? Should you stick it in the refrigerator and just let it thaw over a couple days that way? How would you recommend doing that? And also, maybe you could make a comment about freeze-thaw cycles in your answer.
Okay, so thawing FBS. So in general, thawing them in a 37-degree bath would probably be that would probably be ideal. You definitely don't want to go higher than that in terms of thawing the FBS. When you first take out FBS to thaw, definitely kind of let it sit for a few minutes to kind of acclimate to room temperature. Thaw it in the 37-degree water bath. Let that completely thaw. And then after that, you would want to immediately aliquot the FBS into, workable aliquots, so that every time that you have to remove FBS, you don't have to worry about refreezing and then thawing, then refreezing the same bottle over and over again, because that will have freeze-thaw cycles like that will have serious effects on the quality of the factors that are in the serum.
Yeah, and just to add on that, like when you heat and activate it, you're typically keeping it in the water bath well after it's thawed, like 30 minutes. If I remember the protocol, it's been so long since I've ever done that.
Yeah, I think it's the 56 degrees for 30 minutes. Yeah.
Yeah. So and when you're thawing FBS when it's originally frozen in a water bath, it's kind of like with a vial. You want to the ideal thing, you want to take it out as soon as the ice starts to completely melt away, so it's still a little bit cool by the time you take it to the bench. And like Michael said, definitely aliquot it into your working volumes, so that ideally it should only be thawed twice throughout the entire, when you initially thaw it, and then when you thaw it the second time after you've aliquoted it.
Yep, perfect, guys. That's exactly what I was looking for there. Okay, here's an interesting question. So this asker works with equine endothelial cells, and they're taking forever for her for them to reach confluency. Do you have any advice on how to speed up cell growth in general, but specifically here, how to get to confluence quicker?
So this is a tough one. Like, I would have to kind of see like what the cells, formulations, especially if it's a established cell line that we can like compare against with other records. But in general, if you're experiencing slower growth, one of the more common things to try is increasing FBS by 5%, so that the extra growth attachment factors, growth factors from the FBS can be there in a greater volume, which can help with recovery and general growth of the cells. Other reasons could also be, if they've reached confluency before and you've passaged them, one issue could be that the cells got over-confluent and kind of went back into that lag phase of growth, such that when you passage them to a new flash, they are going to take a little bit of extra time to restart from the lag phase to the logarithmic growth phase.
Okay. No, that sounds good. That sounds good. Okay. Here's another question that's around cell growth. If a cell line suddenly grows more slowly than normal, what could cause this, keeping in mind that they did the mycoplasma test and the test was negative?
So depending on the cell line, it could be something as simple of senescence of the culture. Not all cell lines have been immortalized. Not all cell lines have spontaneously immortalized. And so there are going to be a wide variety of cell lines where after, between 7 to 10 passages, the cells have reached their, doubling capacity, and they're going to senesce and essentially die out over time. So that is one of the more common issues. As you said that mycoplasma contamination was ruled out. Other issues, of course, could just be pH issues, making sure that the CO2 in the incubator matches up with the level of sodium bicarbonate in your media. And also, I guess, checking media too, because complete media most complete media will only have a shelf life of about 1 month. There are other media that will require many more supplements and reagents that will have a shelf life of a week usually. So it may be something with that the media that you're using just may not be able to support the growth of those cells anymore, and you may want to prepare a fresh media to use, see if that helps with growth.
Okay. All right. When storing cells long term, we use our dewar, but we have it full all the time because they thought it was best practice. How full should a dewar be when storing mammalian cells? It's funny, when I first read this, I thought, how full should it be of cells? And I thought to myself, well, if it's ATCC cells, then it should be full to the top. But I guess they need the liquid nitrogen. So
yeah. So most liquid nitrogen tanks should come with some sort of documentation or something saying like 20 gallons for this size tank. A lot of people who companies that do come and refill liquid nitrogen tanks will generally be able to have an idea of like how much would they need for their purposes. They definitely see a lot of biological liquid nitrogen tanks. It's definitely something they would be fairly familiar with, but you want it to be high enough so that it's going to keep temperature. but not like low enough where, you know, you would have to really put cell baskets really far down in them to put them in the vapor phase. But all the way up to the top is overkill, to say the least. You definitely don't want to do that. Yeah, I would recommend actually trying to find any sort of documentation that would have come with the liquid nitrogen tank you have. And if not there, probably try talking to whoever it is that provides your liquid nitrogen to see if they've seen a similar tank and how many gallons that they would use to fill that tank.
Okay. Good answer. Good answer. All right. I don't know if either of you guys have any experience working with triple E. I don't, but this asker was wondering what the difference between triple E and trypsin is in terms of deactivation following use. So triple E reagent is basically another dissociation reagent.
I have not worked with this.
I haven't either.
So I can't say. It says it's free of animal-derived components, so I'm not sure if they're using what reagents they're using.
I do know that that's one of the things that we recommend for some of the HDMI products that we have. Just because I put all those in the back end of our website. But beyond that, I'm not exactly sure. We could definitely get back with you though after talking to some of the folks in our R&D.
Yeah, we could definitely see what we can get from there. I'm looking at the formulation for the triple E, and of course it's confidential. So I assume it's not trypsin, but again, we unfortunately don't have much information about any sort of differences or advantages, disadvantages that could arise when using this versus traditional cell dissociation reagents.
Okay, good. So there's another question about culturing stem cells, such as HSCs. I believe that's hematopoietic stem cells. Do you think the use of RPMI plus 2% FBS is enough to maintain the lineage, or do you need to supply additional cytokines or some additional medium?
I'd have to double check on the recommended expansion medium for these really quickly. If Steve has any information about this, I'm going to just take a quick look and see if I can get an answer for this customer really quickly.
Yeah, I'm gonna let you. Yeah, I'm gonna have to refer to you on that one too. That's a level of detail I'm not familiar with off the top of my head.
Yeah, I would have to check and see the current recommended growth conditions for this. I apologize. I feel like I would definitely have to want to get back to this customer with a more detailed answer sometime afterwards, I would think.
Okay, yeah, that's no problem. No problem. All right. Let's see. All right. Here's an interesting question. Can cryovials expire?
So if you mean expire in liquid nitrogen, no. As long as they're kept in liquid nitrogen, it's accepted that it's, metabolic activities completely cease, so it's pretty much indefinite. They can be, as long as it doesn't get above whatever 130 or I'm sorry, get above negative 130 degrees Celsius or so. But as long as they're in the liquid phase and liquid nitrogen tanks, they really don't expire.
Yeah, we've revived cultures that were 50 years old before, so I would go along with that.
Yeah, like
This is great.
the vials themselves shouldn't have a expiration date, so to say, as long as they're stored correctly. I did see the comment above where about the vial exploding and almost hitting this attendant in their eye. Yeah, so that's not necessarily the vial expiring. What most likely happened there was that the vial was submerged inside the liquid nitrogen, at which point liquid nitrogen can kind of seep into the vial through any cracks if it wasn't tightly closed. And then when you pull it out, that nitrogen is going to rapidly expand, and that causes the exploding vial. So it wasn't necessarily a fault in the vial, more so just the vial being too deep inside the liquid nitrogen.
Okay. All right. So changing gears a little bit, is it advisable to regulate the pH of media with HCI before feeding cells, or once the color changes, should we discard the media?
I would advise against adding, strong acids, strong bases to the pH, to the media. Aside from initial adjustments, for the most part with media, what we tend to recommend is filling the flask with the media and allowing the flask to sit in the CO2 incubator for 15 minutes so that the pH is going to equilibrate, so that you don't have to worry about adjusting it one way or another when the cells are added to the flask. But when we're talking about media color changes, so of course, like as cells metabolize, they create waste. That waste is mostly acidic. You are going to, start to see the color in the media change from a red purple to a orangish kind of color. Around that would be a good time to. That's a good indication that the media that's there has been spent by the cells, and they do require fresh media.
Okay, yeah, that sounds good. That sounds good. And of course, if it changes to yellow and becomes cloudy, bleach it out.
Yeah, that is, any sort of turbidity is not a good sign, and they should be handled right away.
Yep. All right. Once again, let's change gears. Let's talk about proliferation assays, or cell viability assays. Which do you guys prefer, XTT or MTT? And is it because one's more sensitive than the other?
I can address it. Well, the one thing about the XTT kit is it is a bit quicker in that there's a couple of extra steps that the MTT kit has that the XTT doesn't have. Like in the MTT kit, there's a stage where you have to add a detergent to make the cells permeable to the reagent. You don't have to have that with the XTT. And the XTT tends to be a bit more sensitive on the in the area where you take the absorbance and where you read it. You can get the absorbance corresponds to a bit more detailed number of cells. So it's a bit more sensitive, a bit more sensitive as far as getting a more accurate cell number, and it's a little bit quicker. So that's sort of an advantage to the XTT. But they do pretty much the same thing. So both are certainly acceptable.
Okay. We got time for one last question here.
Okay.
When doing experiments with HACAT cells, is it okay to treat the cells 24 hours after seeding it, or should the incubation be longer, like 48 hours? Does the incubation or growth time before starting with the experiment have an effect on the result?
So I can't speak specifically to, this question's specific experiments, but in general, cells that are coming out of cryopreservation are going to need some time. If you're starting with just a frozen vial and seeding them and then performing experiments on them 24 hours afterwards, those cells may not have fully revived from cryopreservation. They may not be the best option to use at that time. As far as other experiments where you have a healthy culture and then you're reseeding it into new flasks in preparation for further experiments, you would probably want to judge by how high of a confluence you need for those experiments, how much, what level of growth you would want to see for those experiments. But in general, just immediately after thawing the cells, I would not recommend starting 24 hours after, because it seems like there may be some more time that is needed for the cells to actually revive from cryopreservation.
Okay, yeah, that sounds good. I mean, that certainly makes sense. And of course, you're going to have to look at this empirically for your specific cell line as well.
Mm-hmm.
All right. Well, I guess at this time we should conclude our Q&A session. I'm definitely overwhelmingly pleased by the number of questions and the quality of the questions that came in this time. And as always, I'd like to thank Steve and Mike for the excellent presentation and the knowledge that they contributed to this session. And thank you everyone for attending. So one final word. We've got a couple webinars coming up. On February 4th, Dr. Tasha Santiago Rodriguez will present her talk on viral metagenomics. And on February 18th, Adria Allen, Alex Picarillo, and Megan Yockey will discuss methods and strategies for viral propagation. Thank you again, everyone, and have a great day.